Hi there, thanks for tuning into this video. My name is Shiv and I'll be presenting you today implementation of mathematical modeling in identifying calcium uptake pathways in cardiac mitochondria. This project or this work was published last year in the Journal of Physiology and was supported by the Virtual Physiological Rat project, about which you can know more by going to this website. I'd like to start off by explaining a few things about calcium signaling pathways and cardiac myocytes in particular. Heart pumps the blood throughout our body. We know that. How does it contract? So there is it's kind of battery of the heart called the synovial nodes, which actually generate the force which propagates through the ventricles of the heart and makes the heart pump blood. So in response to this electrical potential that's generated in the synovial node, there's a surge of calcium that comes inside the myocyte, mostly inside the diatic space, and from the diatic space the calcium basically diffuses to the myofilaments and to the mitochondria. And once it diffuses to the myofilaments generate force through cyclic binding of myosin heads with the actin filament and make the heart contract. But in order to do so, it also consumes energy, and energy is met by ATP hydrolysis, and ATP is produced by oxidative phosphorylation by the mitochondria. So cardiac mitochondria makes ATPs and takes up calcium. It was demonstrated more than 5 decades ago. And over this period of time, as you can see, a lot of more information has been accumulated, and most of it is that a mitochondrial calcium uniporter, that's what they call it, that's a channel, and that's basically responsible for uptake of calcium inside the mitochondria. But these are under experimental conditions. But what happens under physiological conditions when you have all the other ions also present, such as magnesium? So there was a bunch of experiments done by AHA in 2013, and what he did was he isolated cardiac mitochondria and perturbed it with boluses of calcium chloride in the presence of different amounts of magnesium. And as you can see here is that as you go on and increase magnesium, a significant calcium uptake is inhibited. Most of it is inhibited. Or you can see here that there are two components. You can see there's like a fast component and there's like a slow uptake phase. So mostly the slow uptake phase is completely, or most of it is inhibited. Well, so what does it suggest? Maybe there's another pathway that's not inhibited or partially inhibited by magnesium and is rapid in characteristics. Alright, so let's have a look. That's what we did. We try to search and see what can be the possible candidate in the mitochondria. So there are two main candidates that I could think of. One was RAM, that is called the rapid uptake mode of calcium uptake. But it's been shown that it's not at all inhibited by magnesium. And the other one was ryanodine receptor, recently shown to exist and has a calcium graded uptake response. That's what we were looking for in those experiments. And it has also been shown that this ryanodine receptor is sensitive to magnesium, its opening probability is decreased slightly, and its adaptation phase is also its adaptation characteristics are also affected by magnesium. Alright, so that's the one. So we look forward for different mathematical models that's already present in literature, and a model by Kaiser and Levine was selected to be the one because it has a adaptation phenomenon inbuilt in it that was required for our modeling. However, it did not have any magnesium inhibition, so what we did was we added a C2- state to which the model goes to when you add more and more magnesium and sort of inhibits the opening probability of ryanodine receptor. That's what we want. So here are a bunch of model simulations showing how model basically responds in when you add more and more magnesium in the system. So this is a step-up response of calcium, and these are the opening probabilities. And here you can see when you increase calcium, you have higher opening probability, and when you add more magnesium, the rate of adaptation of the channel is affected. And voila! So as you can see here, the mitochondrial model is able to fit the data with that ryanodine type channel. So we cannot say if there's a ryanodine receptor in the mitochondria. However, what we can say is from this demonstration that there's a ryanodine type channel that exists in mitochondria and is responsible for fast calcium uptake. So it took us a bunch of parameters to explain this. 8 parameters were used to fit 30 independent experiments, and we used genetic algorithm for parameter optimization, and the model was implemented in MATLAB, which you can get. All the codes are available on this website, on the virtual rat website. So parameter estimation was done using those experiments done by AHA, and we corroborated the model or validated the model using a different set of experiments which were done in the absence of magnesium. And this dataset was modeled by the basal model that was the basis of our model. But just note that basal model did not account for the ryanodine receptor that we added to simulate AHA's data. As you can see, this model, the model that we developed is able to simulate Blomeyer et al.'s data as well. So it sort of validates that the hypothesis that there are 2 different sort of uptake modes in mitochondria. Well, so what do we learn? So we learned that there are 2 different type of channels in the mitochondria possibly. One is kind of a slow calcium uptake pathway, that's the calcium uniporter, and another one is like the ryanodine type channel, which is the calcium graded uptake and partially inhibited by magnesium, that's the ryanodine type channel. Further analysis of the model actually suggested that the mitochondria are smart. What I mean is that they buffer differently calcium depending upon what the mode of uptake is. So it's just a theory, it's just a hypothesis. It needs to be validated by further experiments, maybe in the future. Alright, so that was the part of the paper, but here I'm going to show you like what the step up should be, because it's good that the mitochondrial model fits the data and things like that, but what do we do with that? So here I used a tracing model, which it's a human heart model, and it does not have a realistic ventricular myocyte, so I plugged in a human ventricular myocyte in it. And further, I inserted a myofilament model which accounts for metabolite bindings and things like that. It's under review and hopefully it will be published soon. And another model, that's the one that we just identified in the previous slides. I couple all these things together and see what happens when I knock out the ryanodine type channel, which I call MRYR here. So once you knock this channel out, you can see the calcium uptake is not much in the mitochondria, and therefore you have a surplus of calcium in the ventricular myocytes. Alright, so what's the consequence of this? So as a result of this increased calcium, you can see there's a minute difference in the Frank-Starling curve. What basically it shows you is that as the filling pressure increases, how the cardiac output changes of the heart. So your cardiac output is somewhere in between, as you can see, is that it's relatively unchanged. So bad news? I would say so, because as you can see in the mitochondria, the uptake in the absence of this fast uptake channel is significant, and we are not accounting for it. What I mean is that mitochondria, as I said, is responsible for the production of ATP that provides the energy for contraction of the heart, and which I'm not accounting for, like how this calcium is affecting myocyte. That's not been done here. It's a work in progress, and hopefully in the future we can see if there's an effect of mitochondria, and how significant is that. That's for later. Thanks guys. Thanks so much for staying and listening up. And if you have any questions, shoot a mail to me, and hopefully we'll talk to you again later. Ciao.